I'll ask you please to just open the curtain and leave the translucent filter, and we will continue with questions from the audience. Any questions to any of the speakers? Two questions here. We have to be very brief.
This is a question for João. You show that A-beta increases the cleavage of the TKRB receptor for the full length. And I wonder if there is any role for the ICD fragment that is released after the A-beta activation.
Sorry, you are asking about the function of TrkB ICD fragment, right?
Yeah, if there is any function for the ICD fragment.
Okay, so until now what we are doing is trying to understand what is doing on nucleus, where it can be a repressor or a promoter in the genetic expression, and we will do that in next few months. We are also trying to understand which proteins are being phosphorylated by TrkB ICD fragment, and what is the impact of ICD upon cell viability, because it is known that other calpain fragments impairs the cell viability and so leads to neuronal death. And if we could blockade TrkB full-length cleavage, we could prevent two different things. So one is the effect of TrkB ICD fragments, and another is the maintaining the BDNF signaling intact and functional. And concerning the characterization of TrkB ICD fragment, it is just these 3 or 4 parameters that you are attending.
Okay, thanks.
Thank you for the speeches. Wonderful, all of you. I have a very specific question for Cláudia, because I'm working on being one. Yeah. And my point is, you know, maybe it's due to the time, but I miss in your speech the context of other researchers working on being one and the findings they have compared to yours, because in some ways it looks to me that are in the opposite direction, specifically the Jean-Charles Lambert paper in molecular psychiatry.
I cannot understand very well, sorry.
Can you repeat the question?
It's just I couldn't understand very well, sorry.
The question is, you know, if you can put in the context of the previous paper related to the function of being one, your findings, the Jean-Charles Lambert paper in molecular psychiatry 2013 indicate a correlation with the tau pathology, not amyloid pathology, and overexpression is related to the allele we detected in 2010. So what is your opinion about that findings?
Right, I mean, I think it's like with, in terms of the correlation with tau pathology, even in the patients, we know that tau pathology correlates better with disease progression than amyloid plaques. My point concerns what triggers the pathology, so the first initial changes. And I think those are accepted for more than less, that it's the A-beta accumulation that then triggers those things. But we cannot really measure these all species that are really the most, the earliest toxicity. The oligomers are not really used to measure correlations. So in that sense, I think it's the fact that BIN1 correlates with tau toxicity reinforces the role of BIN1 as a risk factor for Alzheimer's disease. So BIN1 had been studied before as having a role in endocytosis, and what we show is that loss of function of BIN1 potentiates A-beta production. So I think everything makes sense to me. Thanks.
One final question.
I have a short question for Peter. How do you explain you find different amounts of alpha-synuclein in LRRK2 and idiopathic patients? Do you have any hypotheses or?
I would like to have the explanation. Unfortunately, it's still in this moment, it's just our observation. It's a descriptive study. The only conclusions which I can say is the conclusion I already mentioned. It's just a indicator of a different underlying molecular mechanisms. Unfortunately, I'm not able to provide a good explanation for this. Could be that it's also related with the mitochondrial biology, which we're studying mainly. Could be some independent pathway. Unfortunately, this is the...
Thank you very much to the speakers. It looks like we're getting a very broad range of mechanisms. I am not from the field. It's hard to make sense of it all. There's stress, there's oxidation, there's inflammation, genetic predispositions to trafficking. And as we continue on the summit, we're getting a larger picture on what may or not matter most, how these things interact. Thank you very much for your contributions and for your talks this morning.